So in your brain, we think there's something like 100 billion neurons. And we think on average, each neuron makes connections with 10,000 other neurons and then receives connections from 10,000 other neurons. So if you multiply those numbers together, you get something like a quadrillion. 100 billion times thousands, 10,000 gets a quadrillion. And that's a hard number for me to keep track of. One way to think about it is that is 10 times more stars than there are in the Milky Way galaxy. So every human has something like 10 galaxies worth of neuronal connections in their brain.
My guest today is Dr. Narayanan Bobby Kasthuri.
Yeah,
Thank you for joining me.
Thanks for having me.
You are a neuroscience researcher at the Argonne National Laboratory and also an assistant professor in the Department of Neurobiology at the University of Chicago.
Technically, I just got tenure, so I'm now an associate professor.
Oh, sorry.
No. I haven't updated any of the online web,
right.
mostly because I get to say that. It's a cheap way to talk about myself, I suspect.
But so as I was preparing for this podcast, I wanted to try to get the pronunciation of your name correctly.
Yes.
Narayanan.
Yes.
And I discovered that is quite a big name.
Yes.
Lord Vishnu.
Yes.
The preserver and protector of the universe.
Yes.
That's a tall order.
I know, and in fact, other than being named one of the three principal gods of, if it was up to me, I should have been Shiva, the destroyer, because I think it suits my personality, at least my scientific personality, a lot more. Yeah, and so not only did they give me the name of a god, which didn't help, they gave me the name of the wrong god.
But how did Bobby come about then?
So there's two versions, and I'll tell you both if we have time.
Yeah.
So my dad loves telling this story, and you may, I'll tell you ahead of time, you may have to edit parts of it out. But I was born in a tiny little village in India called Goa, near the mountains.
It's not so tiny anymore, is it?
Not anymore. I guess, yeah, fair enough. Right. Back at the time, it was a tiny place. And my mom and dad lived there. And my dad loves to tell the story about how the nursemaid, the local nursemaid in the village that we grew up was also the town prostitute, which he says explains so much about me. And in one of the dialects that she speaks, and then the second thing he says is my mom had trouble breastfeeding, so I was raised on goat's milk, which he explains, he says, is why I'm so hairy. Growing up, my dad has a wonderful sense of humor. And in her dialect, the word buppy means little goat. And so she would call me Buppy growing up because I grew up on goat's milk. My parents started calling me that. And then when I came to the United States, Narayanan was the name, but my friends noticed that my family was calling me Buppy. And the closest version of that was Bobby. And then they just took it. And then I took it. And then people would ask me, and I think I lied to them, which shows you even at that age, because I didn't want to tell the story of the goats and all of that stuff. And they'd say, why are you named Bobby? And I would say, well, I'm named after my favorite character on the Brady Bunch, which was this TV show that I watched obsessively growing up. And there was a Bobby Brady on it. And so that's apparently the origins of my name.
Two good origin stories.
I like guess, that. Right.
So you work in a relatively new field of neuroscience, correct? Connectomics.
Correct.
Could you explain what connectomics are?
Yes.
So
the broad idea is that, and just a little background, brains are almost entirely made of these cells called neurons or brain cells. And there are at least two ways that neurons and brain cells are different than any other cell in your body. A stomach cell, a liver cell, skin cell, any other organ. And those two ways are, one, neurons are no longer divide after you're born. So every other cell, and I'll get back to this in a second, every other cell in your body winds up getting replaced. So over an interval of like 5 or 10 years, you don't have the liver you had 5 or 10 years ago because every one of those cells has been replaced. whereas the brain we think that you were born with is the brain that you died with.
Yeah.
That's the first difference. The second difference, which is even more obvious, is that almost every other cell in your body can kind of be described geometrically or morphologically as little spheres or cuboids or et cetera. But neurons do this remarkable thing, which is that they send out parts of themselves called axons to connect with neurons, sometimes on the other side of the brain, sometimes down in your spinal cord, and they make very, we think, specific connections with certain other neurons in the brain. But the numbers are kind of astronomical. So in your brain, we think there's something like 100 billion neurons. And we think on average, each neuron makes connections with 10,000 other neurons and then receives connections from 10,000 other neurons. So if you multiply those numbers together, you get something like a quadrillion. 100 billion times thousands, 10,000 gets a quadrillion. And that's a hard number for me to keep track of. One way to think about it is that is 10 times more stars than there are in the Milky Way galaxy. So every human has something like 10 galaxies worth of neuronal connections in their brain. And the goal of connectomics is to ultimately map everyone, to know for every neuron its 10,000 partners that it sends information to and the 10,000 partners it receives information from. Because the strong hypothesis in neuroscience is that that map of connections is unique from animal to animal, especially among mammals, and that map of connections contains everything about you. Every memory, every skill, every fear, every dream is somehow associated in that map of connections. So the field of connectomics, I guess sort of self-explanatory, is to develop the technology to start making those maps. That was a long answer. Sorry.
So how do you even tackle a challenge so massive?
Yeah, I mean, I think the first thing is to sort of get the idea of the constraints of what it is that you want to try to do. So the first thing is the only way you can put a quadrillion of anything in a volume this small is to make each neuronal connection extremely small. And the first thing we thought about when we were thinking about tackling this problem is that those connections are so small they cannot be visualized with standard optical microscope, regular microscopes that you find. They cannot be visualized with things like MRI or others. So we realized that only sort of a kind of microscope called electron microscopes that have resolution orders of magnitude better than an optical microscope can actually visualize these connections. So first we realized, we have to use microscopes that have nanoscale resolution, like electron microscopes. And then the second constraint that becomes obvious is that when you take something that's nanoscale and you image something that's this big, you're going to produce data sizes that are larger than humans have ever produced in the history of humanity. And then the second corollary is that you can't take your time collecting all of that data. So not only because it's so much of it, we realized that we needed extremely precise microscopes like electron microscopes, and we needed them to go extremely fast if we wanted to map brains in some period of time. And then the third question, which we are still dealing with, is what are we going to do with all the damn data? But those were the kinds of three constraints that helped set up the problem maybe 15 years ago. And then there are lots of little things that fall out of it. If you want to prepare electron samples for electron microscopes, you have to section them really thin because the electrons don't penetrate very deep into your sample. You have to label them with heavy metals because that's what gives you a signal, the same way fluorescence gives you a signal in optical microscopes. And so a bunch of sort of practical issues came about once we decided that these were the three big constraints. I don't know if that made sense, Paul.
Yeah, definitely. But so if, let's say this is the human brain here,
yes,
then you're slicing it. Does it matter which way? I assume it matters which way you slice it.
That's a great question. Right now we haven't thought that the orientation matters. Mostly because, and this isn't exactly true, but in many parts of the brain, neurons go in all directions. They send their axons in all directions. It would, if brains were really well laid out, then we would have an orientation. Maybe it's best to cut along the axons. Maybe it's best to cut the axons orthogonally. But since we think they go in every direction, we haven't yet settled on a particular orientation.
Yeah, and when the neuron sends a message, is that unidirectional? Is that multi-directional? Can it send and receive?
Yeah.
Along the same axon?
No. So one of the advantages we hope of mammalian systems is that they're very simple. So a neuron has two parts. It has something called a dendritic tree, which sticks out one end, and it has a very long cable which we call an axon. The axon is essentially output one direction, and the dendrite, to a first approximation, is input one direction. So we hope that, that being said, we know so little about brains that someone watching your podcast will say, there's this neuron in this part of the brain that might send information. But to a first approximation, we think it's all one direction.
One direction. That's interesting.
Thank God. Because if they added that, it's bad enough to have to map 100 billion of them, but not to know which direction they're going is really going to make the problem.
So, do you have any idea of scale of time? Like, how do you think it's going to take a decade or two decades? Or?
Yeah. So first, before I answer that question, I think, and I keep meaning to look this up, there's a very famous quote that says, humans always overestimate what they can do in a year and always underestimate what they can do in 10 years. So we should just take that a grain of salt. What I would say is that previously, the animal where we had the entire wiring diagram for was a simple worm called C. elegans, which doesn't have 100 billion neurons, has 302 neurons, and it's extremely small. Somebody in the 1970s and 80s mapped all of those connections by hand, and it took them years to process, to analyze, to look at those samples. After 10 years of building up the technology, we can now do that in weeks. And in fact, very recently, somebody just mapped the fly brain, which was not 302 neurons, but closer to 1 million neurons. So something that really...
The fruit fly.
The fruit fly brain.
Drosophila.
Thank you. Sorry, the fruit fly brain. And that took, 5 years and some amount of money to do. But we estimate now that the mouse brain, and I have some samples of mouse brains embedded in plastic for you if you'd want to look. Here's another version. We think is probably done, hopefully in the next 5 years. And that's 100 million neurons.
100 million.
100 million. Still not the human. So if I had an estimate, I would say more than 10 years out for the human.
10 years out.
Yeah, but not 50 years. I really would like to get it done in my lifetime.
Now, I'm assuming that you are working in conjunction with other other scientists and then you bring all your, so as not to duplicate work, are you all taking different parts of the brain?
So sort of. I would say that the field is still young, so we're still frenemies. I don't know if that's still a word that people use. I've known them, everyone, and they're wonderful people. But each of us currently has our own version of that electron microscope that we think is going to be the fastest, best resolved electron microscope. We have our version, our competitors' friends at Harvard have their version, our competitors' friends at Princeton have their version. So right now, before current events, the idea was that the NIH was going to sponsor a bake-off. They were going to they've given us money and some time, 3 to 4 years. Show me the best thing your microscope can do, show me the best thing. And then the idea would be that in 3 to 4 years, we would decide on which of these is the best version. And then we would take that microscope, subdivide the brain, and do exactly like you said. Very similar to how the Human Genome Project was done. The first thing they had to do was settle on what's the right kind of DNA sequencers. And then once they settled on it, they had many places, each of them working on a chromosome. So WashU in St. Louis was working on chromosome 12, Columbia was working on chromosome 18. We think there's a similar strategy for the brain.
But there's that much variation in electron microscopes in terms of their...
So I wouldn't say there's dozens of versions, but there's 3 or 4 versions. And then the optical people still keep trying with their super resolution techniques, et cetera. So yeah, I would say maybe there's a half a dozen viable approaches, but not dozens.
And how many years are you into this process now?
I've been doing this for about 15 years. Our version of our newly discovered electron microscope, we've been dealing with for about 2 years.
Okay. So another 8 years.
Yeah, I'm hoping 4, but I'd settle for 8.
So what models do you use in your research?
So mostly we work on mice, and that's mostly because the mouse is probably the workhorse of neuroscience. It's the model organism used by the most scientists. So for a long period of time, even today, we wanted this approach of connectomics to be something that was applicable to many scientists. So we started working on mice. It is worth saying that the approach works on any species, and the thing I'm particularly interested in is this idea of and I take a little tangent. Sorry. And let me just say it the way I tell the students, and so I apologize. There's only been 2 great ideas in biology. I'm going to make the argument. The first is the idea of evolution, which I think is still the most profound idea. And then a second idea was the idea of tying development to evolution. And it was popularized by a scientist who passed away named Stephen Jay Gould, who was one of the great evolutionary biologists, and he wrote a book called Ontogeny and Phylogeny. And just to explain, ontogeny is a very fancy word for development, and phylogeny is just a fancy science word for differences across species. And he didn't, but he popularized this idea, and it really stuck in my brain because I think it's the second most important idea, which is that all differences in species have to come from developmental programs, because we all start, at least among mammals, as a single fertilized egg. And so somewhere between fertilized egg and a primate brain, there were a series of changes, a series of steps, and somewhere between mouse, fertilized egg, and mouse brain. And if you could find the moment where those developmental programs diverge, then you're on to what makes a primate in terms of cognitive abilities, and in my case, hopefully, what makes a human a human. So although we've been working on mice, it was more to set that system up so that we could ultimately compare ontogeny and phylogeny across many systems. And in that world, my 3 interests are mice, non-human primates, and humans.
And I was looking at one of your papers that was looking at the brain of a mouse compared to that of a
primate.
A primate, a
Right.
macaque.
Macaque.
And you found that the mouse had more neural connections than the primate.
That's right.
Which would be counterintuitive.
Correct.
But then I saw that
Yes.
later research,
That's right.
It gave an explanation as to why that may be.
Yes.
In terms of, so you were looking at an adult mouse brain versus a juvenile, or no?
Yeah, so excellent. And
juvenile primate.
The most important thing about science is to say when we made mistakes. And this is a classic example of it. So we did the first paper where I just was curious, and I believe that a primate adult brain is in some ways more sophisticated, if I'm allowed to say that, than in a mouse.
I think most people would think
would
that,
agree,
right?
right? So what we did was we chose 2 time points where the field sort of said this is the equivalent of an adult mouse, and this is the equivalent of an adult primate. And the way the field does that is it's fairly simple. We take the life expectancy of a mouse, which is 2 years old, and we take the life expectancy of a primate, which is 20 years, and we say, okay, what's the relative time. And so for a mouse, we thought that would be 1 to 2 months, and for a primate, a couple of years of age. So we compared those brains, and it turned out that the mouse had more connections per neuron than the primate, which really threw me, because I thought, to a first approximation, having more connections gives you more sophistication.
Yeah.
You're connected to more neurons, you have more options for the information to flow. So that bothered me. And so we decided to do a second experiment, because something implicit in that argument about the adult is that somehow the idea of development scales to the lifespan. So for example, again, to take a mouse that lives 2 years of age, you figure that the brain developed in a couple of months, because development scales to the lifespan. Whereas a primate that develops for 20 years, maybe it takes 2 years to get the development done. And that's a fairly intuitive explanation. And so we decided to test it, which is where we figured out the mistake we made in our first paper. And the result we got is that development, at least of neuronal connections, seems completely independent of an animal's lifespan. So that a mouse takes as long to develop its connections as a primate does. So you can't just choose 2 months of a mouse and 2 years of a primate and somehow assume those are both adults. In fact, they have the same clock. So all we did was compare we think now, a very juvenile mouse to an adult primate. And that's how the results were sort of confusing.
Yeah.
Did that make sense, Paul?
Definitely.
Good.
Can this be extrapolated out to other mammals then as well?
Yeah, so we didn't do the work, but we then were like, wait, this is weird. So we went and checked the literature for all the developmental of connections, not all of them using our advanced techniques and et cetera. And we found, at least as best as we could tell, that for every species except for 2 that has been studied, that's rats, cats, different kinds of primates, they all seem to have the exact same clock. And they all have rabbits, they all have different lifespans. The 2 exceptions we found in the literature were chimpanzees and humans. And weirdly, it turns out that one of the theories of what makes human brains special is this idea of neoteny. And what that means is that human brains take forever to grow up. First, we're born, I have 2 kids, as I was telling you, and both of my kids, they hate when I say this, we're just born useless and helpless and hapless. And I think it's been a common fear among parents across the history of humanity.
Yeah.
Holy crap, these kids are gonna die if I don't take care of them.
Right.
100%. And most species aren't born that way. Most species are born knowing much of the behavior they need to survive. But humans have this weird, it takes us a year sometimes to learn to walk and talk and et cetera. And so there is this idea that what makes our brains special is that they're so slow to develop. And because they're so slow to develop, it allows the world, the environment of the world, to shape that slow development, which is why humans succeed in the tundra versus the desert versus urban cities, et cetera, because we have this capacity to learn from our world. What our results suggest is that neoteny, that delayed development, is a quantal invention. It could have been graded. It could have been that a mouse develops very fast, and a primate is 30%. A rat is 30% slower than that, and a rabbit is 30% slower than that, and primates and so you had this kind of smooth curve of developmental delay that reaches to humans. But instead, I think, much like human intelligence, everybody is packed into one curve. And my suspicion is humans made an hominids, which is chimpanzees and humans, sorry about that, made some quantal leap in delay. And that's where I am now.
But no one's looked at marine mammal brains then.
Yeah, they're hard to get. We did try to work with the Shed Aquarium, but they're also Many of them are endangered, the ones I'm interested in,
Right.
and people aren't ready to give us their pups yet so that we could track development. But that's an excellent question.
Yeah.
And even not and I'm not even interested in those, just in those, but there are large elephants. What I'm really now, to back out the argument, another way is that there are mammals other than humans that spend a long period of time nurturing and teaching their young. And I suspect this delay in development and the period of which parents take care of their children are fundamentally linked. And there are animals like elephants and dolphins and whales that we both consider intelligent, but also spend a tremendous amount of time raising their young.
Right, yeah.
It's really hard to get those samples.
What do you think is the most controversial thing happening in the field of neurology currently?
I don't find it controversial, but often when I talk to students, it comes up again and again and again. At least from a neuroscience perspective, there's very little space for free will. I don't, like, all that's up here is just neurons and connections. So maybe this is still controversial, but there's very little space for the soul, which I don't find controversial at all. But in reality, this network of neuronal connections, there's very little space for free will.
Yeah.
And even more weirdly, and I know this is all well known, it's just hard for people to get their minds or brains around it. There's very little space for you as an individual. There is no Paul neuron. There's not even 5 Paul neurons. Paul is distributed over 100 billion little machines that are following machine rules. Inputs mean this, inputs mean this. And somehow that creates this illusion of me that's unified both in space. There's a single me in here, and I've been the same me unified in time. I think neither of those things are true. And I find, and usually I tell the argument, it slowly is leaking, that idea is slowly leaking into our everyday lives in society. I give the argument that, 30, 40 years ago, if you had a DUI, you were weak-willed. You lacked this kind of willpower or free will. And the legal system took action based on that worldview. Now, I think when you get a DUI, there's this idea that it's a biological problem or a biological situation, perhaps beyond the free will of that particular individual. So therapy, medication are the right ways to think about this. And I think that's the world where it's going to be more controversial to think about what culpability, personal responsibility, these things mean, the more and more we map a brain.
So that reminds me of something else I read, one of your previous papers too, looking at the effects of cocaine on a mouse brain. So it causes a rewiring.
Fast.
But is that reversible?
Yeah, so we didn't check. We'd love to check. That's the question.
All right.
That's the question.
So that's still
That's what we're working on now.
Oh, it is working on that.
We're a little, not so much with cocaine, but more with opioids now, only because we thought, I'd like to know the answer, and this answer is critically important for our country. So that's where we're moving that research.
Right.
But excellent question. And just to push this, if cocaine makes your brain physically rewire, and that physical rewiring leads you to need more cocaine, not that you're a cocaine addict, Paul, but if you were, where's Paul in that world? What's his culpability? What's his responsibility? I think is where the controversies are really going to start.
This is another tangent
Please.
here, but I'm looking here at your Einstein.
Yes.
Would it be possible that someone of his intellectual stature, would they have more neurons than the 100 billion that we're talking about? Or
Yes. Although it's true that the variation, there is variation. When I say 100 billion plus or minus 20 billion,
Yeah.
it isn't obvious that that's the number that changes between Einstein and me, for example. But again, I will say the thing, when people asked Einstein, what was it, where does your genius come from? And he said, look, I've always been able to visualize ideas as a child, almost as if his development was even more delayed relative to human development. And there's this idea, I don't know how much of it is apocryphal, that he didn't do well in school. He was a sort of a slow student. He's a late bloomer, I think is what people say. And there's this idea that a lot of genius is associated with late bloomers, and in his particular case. So I'd make the argument, with very little data, that it's not the number of neurons he had, but it was his developmental trajectory.
Yeah.
And weirdly, slowing that developmental trajectory wound up in his genius. The other way to say it is, I often hear of the other opposite extreme are prodigies. Kids who seem to have accelerated intelligence, primarily in music and math, I think, are the
Like a
two.
Mozart or something. Yeah.
And in fact, that's an excellent point. Will you pin that, please? Because I think he's the classic experiment that we'll never know the answer to. And my understanding of the research is that the interesting thing about prodigies is that they're ahead of everybody at a certain age, but at some age people catch up. And it's not that they can maintain that early development out into life. The one I don't know, because he died young, is Mozart. He seemed to be such a prodigy, and such that, is it true that if he had lived to his 50s and 60s, the rest of the musicians
Would have caught up
with him? It's hard to believe, I guess. I'm not a music buff, but given how amazing the music was. But I think, perhaps this is controversial, I'm trying to raise the tension that early achievement or early achievers are not the pool that we get genius from. It's the late bloomers. Now I'm biased because I have this idea of delayed development.
Right. So we believe that the human brain doesn't fully develop until the age of 25.
Maybe even later.
Maybe even later.
That's right.
So in someone like Einstein, maybe that was 35.
Exactly. Exactly.
Okay.
And in fact, there's often this idea that for different fields of science, you have a window. If you don't get great math done by the time your 20s are done, you're probably never going to do great math. If you don't get great physics done by the time your 30s are done, you're probably never going to do great physics.
Right.
And what's interesting about him, apparently, is he continued to do these great physics experiments into his 40s, and so consistent with this idea that his curve was somehow right-shifted or delayed relative to others.
I was also reading that, so the brain, it develops from back to front.
Allegedly.
Allegedly.
Yes, which is also how we think of the cognitive abilities of the brain. The back of the brain is not simple. Let's say the cortex, which is the part that's evolved the most in humans. Back of the brain, we call primary visual cortex. It's just seeing shapes and lines and et cetera. And the front of the brain is where we have executive functions and decisions.
Right.
So it also seems consistent that there's a developmental delay within a brain related to how sophisticated the brain region is.
So would the back be considered the lizard brain? Our lizard brain, or no?
No, so I would, the lizard brain probably are the subcortical structures, places like the amygdala or the basal ganglia, or et cetera. It tends to be mammals and a little bit birds where cortex evolved. But it is probably fair to say the oldest version of cortex is the back of our brain, and the newest version, the part where evolution has exploded it in humans, for example, is the front of our brain.
Yeah.
I used to think, Paul, I have these ridges here, and I always used to tell people, this is because I squeeze so much frontal cortex in. And then we all, at some point, we had a collaborator here, all of the lab got MRI scans done, and it's just air. It's just empty. The lab did not let me live that down, man.
Just air. How do you see the field evolving within the next decade?
So I have ideas about both neuroscience and connectomics. Two things, I think. One, I think we'll have to give up our idea that the way to understand brains is to do standard hypothesis, serial hypothesis testing. I think maybe that was useful for physics and chemistry and maybe some simpler systems, but the brain is so recursive and complicated that we're going to have to kind of abandon the normal scientific process of trying to investigate brains. And I think the field will evolve to, like we were talking about, how genomes evolved and how proteins evolved. We're going to make digital representations of brains. Maybe we'll map an entire brain, upload it into a computer, and let AI run the experiments on that computer. Let it do the millions of experiments in parallel, manipulating all of the other strategies. And I think ultimately, then once it gets an answer, it's going to have to really dumb it down for us to understand it. Let me say it a different way. I have a couple of cats at home, the animals. And I hate them. They're idiots. They're the bane of my existence. And my family obviously loves them more than they love me. And I'm convinced they're so dumb they will never understand how a brain works. Is it obvious that a human brain should? And I understand that some human brains have done remarkable things understanding the smallest parts of the universe, to the black holes, to galaxies, et cetera. But I don't think it's infinite. And I think there's something really complicated and sophisticated about a brain that I think ultimately it's going to require AI to answer it. The same way it required AI to understand how proteins folded, which they just gave a few Nobel Prizes for. That's where I see the field going.
Nice.
It's a little depressing for me.
Well, so that leads me to this question then, what led you to study the human brain? Is it simply the complexity that intrigued you so much?
A little bit. It's always nice when you're talking to other and you say, "Oh, I studied the brain," and you get to pretend like you're smarter than them. Not like we made a ton of progress. But for me, it was so. Just to say, growing up, my parents Indian. I'm Indian. And so there was this strong idea that Indian kids should go to medical school, which is why I wound up in medical school. I wasn't sure I wanted to be a doctor, but my family does have a history of mental illnesses. So I really thought, okay, I'm here. Why don't I see how I can help mental illness or understand it a little better? And although this was 20 years ago, I think the answer is still the same. The smartest people in the hospital are the neurologists, the psychiatrists, the neurosurgeons that I met. And despite that, we have almost no sort of rational treatment for any of the brain disorders that we have. And it was almost this moment, it must be narcissism when I think about it, where I was like, well, I went to medical school, I did what my parents asked, I'm trying to learn about mental illnesses so I could go back and help people, but this is kind of a joke. We don't know anything. And it looks like I gotta do this. Otherwise, I don't want to do this. I'd rather be a high-paid neurosurgeon, but the problem doesn't get solved. And so I was like, okay, screw it. I'm just going to figure out the brain in about 5 years, and I'll be back to neurology.
So then you went back to school for your PhD then?
I had my PhD already.
At that point. That was before medical school?
It was during medical school. So it was somewhere, everything was happening at the same time. Fair enough, I probably took that PhD because I was so, I don't want to, it's not like no one's not trying as hard. It's no one being lazy, it's no one being stupid, any of that. It's just, it occurred to me we just needed more people working on figuring out how the brain works if we were ever going to figure out how the brain didn't work. And then, because I'm, again, something, narcissist or whatever, I read this, my other hero who will put up, my Illinois hero is Abraham Lincoln. I was saying about
Oh yeah, You never told me. Did you end up naming
Oscar.
your son?
Oscar.
Not Abe.
And I said, not him. My wife was like, this is insane. And somebody once asked Lincoln, how do you navigate something as complicated as the Civil War? And I thought of the same, how do you navigate something as complicated as a brain, as understanding the brain? And Lincoln's answer, which is just genius in the way I think of him, is he said, oh, that's easy. Just work on the hardest possible version of it and ignore everything else. And when it turns out that if you can or cannot do the hardest possible thing, then you know. And if you can't do it, okay, try the second hardest possible thing. And he said, that's the path to success. And I thought, okay, what's the thing I didn't think it at the time, that I could spend 15 years of my life, and that was to make this map of neuronal connections. And every year I get enough of a taste that it's going to succeed that I'm in now 15 years into it with the same strategy.
To keep going.
Yeah, exactly. In fact, just to say, that's not even a Lincoln quote. It's so the other thing I'm really fascinated by, even when I was, that's the original New York Times from the day that we landed on the moon. And it's the only time in history, I think, that the entire New York Times was devoted to just one thing, one news story. And when I was young, I learned this idea that when we land on the moon in 1969, the average age of a NASA scientist was 29 years old in 1969. Kennedy makes his big speech about why we go to the moon 7 years earlier, in 1962, which means most of those kids were in college. And so I really believe this idea that big science moves the needle forward. That if you can do a big project, whether that big project succeeds or not, what you do is you engender a whole new generation of scientists to join science. So I wanted to do something hard, and I wanted to do something big at some scale. Yeah. Because then if I fail, which of course the idea we're going to map the brain and understand it, my actual path to success were the hundreds of people I convinced to become neuroscientists, and I'm happy to take that as a success.
That's where your name fits you then.
Fair enough, maybe. Fair enough. I never made that connection. Yes, but thanks you. And so that's what sort of led me to this idea.
How do brains grow and age then?
Yeah, I don't know.
We don't know.
Sorry. So just to get back, as I was saying, there are two fundamental things about neurons that are different than every other cell. The second one we talked about, the axons. So now I'll get back to the first one. That is that at least for every other cell in your body, age is determined by the number of divisions the cell has. In fact, allegedly there's this piece of DNA at the very end of your chromosome called the telomere or so.
Oh, right, sure. Yeah.
And
telomere length.
And telomere length, which shortens with every division, is aging. Meaning that's how my liver cell ages, because it divides. Telomere length shortens, it divides, and then at some point the telomeres get so short that the cell starts to lose its ability to function. So that's the model for the rest of the body. But as I said, neurons don't divide. So I don't know why people haven't thought about how is aging determined there. At least our general model of aging is each division is the equivalent of a ring in a tree. So if you could calculate the number of divisions, that's the age of a cell. But in cells that never divide, what does that mean is the question. Now there are cells in your brain that do divide. There's the blood vessels, those cells divide. There are these other non-neuronal cells called glia. They divide. So perhaps those are the cells that are making the brain age, and neurons are just eternally young.
No,
I don't know if that made sense.
Yeah. With the human brain, obviously it's a very energetically costly organ, right? I think I read somewhere it uses 20% of the energy.
That's right. Maybe even more.
Maybe even more.
But it's only, say, 5% of the mass. So it's some 4 to 5 times more energy per mass than any other organ in your body.
Do we have comparative studies, like how energetically costly is it in elephants, let's say?
Great
or
question.
We don't know.
Great question. The idea would be that all brains, so it's interesting, just to push on this, in one sense, brains are incredibly energy costly, exactly like you said. They take up a bigger fraction of the calories or sugar in your body than they deserve to based on mass. But another way to say it is they're incredibly energy efficient. Now, 20 watts is, these light bulbs probably are more than 20 watts of energy. And these new AI algorithms that we hear about from OpenAI, et cetera, they want to build their own power sources, their own nuclear power plants to get that AI to do the same thing that a human brain does for 20 watts. So I don't know exactly, in terms of the body, it's a huge energy sink cost. But in terms of what it achieves, it's incredibly efficient relative to our computers and AI currently.
Right. Well, then, to go back to the Einstein
Yes.
thing, then, could we theorize that his brain was using even more energy, or the opposite? Was it using less because it was more efficient?
Yeah, and so just to give you a story, it's such a sad story. Shortly after Einstein died, a neurologist stole his brain and went on a cross-country trip with it, and damaged it and brutalized it in ways that just, what a shame. And so we will never actually know what the architecture, if there was anything special to the architecture of Einstein because of this crazy cop chase across the country. But the one thing that people have said is he had a higher ratio of these support cells called glia to neurons than you would expect in a normal brain. And what glia do is control one of the things glia do is control the energy in the, so there is at least some, exactly what you're saying, there is at least some suggestion that his brain was more energy efficient because he had more of these glia to control it. But I don't know how to think about these results because they're so potentially artifactual.
Right. And then someone that would have a low IQ then would be using a lot more energy then.
That's right. We're really speculating here, but that's the idea.
But no one's looking at that at all currently.
No, in fact, I'm not sure how they measured the first, usually they look at heat. I think they'll take an infrared map of a brain and look at heat dissipation and use it as a function of energy consumption. But let's write this paper. Let's do a paper together.
Let's do it.
Because I'm not sure whether anyone has done this for a primate brain, for a mouse brain, whether across species or development. I'll ask AI later to search the internet for it. I like this idea a lot. It's an excellent idea. Excellent.
Maybe this is not a good question, but one would think that each human brain, right? Your brain is unique and different than mine.
Let's hope so.
Or is that not the case?
So this is an excellent question. So usually when I describe this, I say it's possible, it's likely that every brain is a snowflake. And it represents your unique history, your experiences, the things you were exposed to either in the womb or afterwards. But even snowflakes have a common theme. They all have 7 arms, or I get spiders have 8 and whatever, but 6 arms or something like that. So there is some structure on which there's a uniqueness superimposed on it. And like I said, it is true that the back of your brain is responsible for vision, and the back of my brain is responsible for vision. So there's some commonalities or some template that we all share. But at the same time, your personality is probably different than mine. So a question is, what are the parts of brains that are truly unique across humans, and which parts are the same? And I don't know how to answer that question until we get the full kind of brain connectivity map. So we can actually ask that question about the connections as opposed to this neuron or brain region.
So that's why this map is so important, right?
Yeah, I mean, I believe it, but I'm also the guy making the map, so I'm a little biased to say that I think it's one of the most important data sets that we can get.
That being said, we would have to map multiple brains
Absolutely.
then too, right?
Absolutely.
To have some. Yeah.
Absolutely. I mean, the goal is to get to one, but again, the goal of the Genome Project was to get to one.
Right.
But once we got to one, people invented so many new technologies that made it 10 times, 100 times cheaper, and now we're at $1,000 genomes, or maybe even less. And so that's the kind of inspiration that we take. The goal is never to. The golden-plated individual brain will be very useful for lots of things, but not that useful. Really, it's thousands of those where I think the real utility will come in.
Yeah. What is the biggest misconception people have about the field of neuroscience?
My colleagues are probably going to get mad at me. I think people think we understand things like an order of magnitude about the brain than we actually understand. There are things we really do know about brains, about how individual neurons work, about the receptors and the channels inside them. We know a tremendous amount about that. But somehow coming to theories about how brains move or think or sense, we are really far behind. I sometimes would say that we are pre-Newtonian. We don't even have the 3 fundamental laws of brains yet. And so I think it's amazing to say, well, I shoot lasers and I have electron microscope and I've done all of this. There's 50,000 people working every year, 100,000 probably on brains, the smartest people I've ever met. And then people have this idea, well, okay, then you're probably just a year away from Alzheimer's. You're just a couple of, and I think it's hard to describe how profound our gaps are in understanding brains. We don't even know if many of the results we have are truly causal results, like this causes this thing to happen in brains, or just correlational, because it's so hard to experiment in a very precise way. And I think this is why my colleagues are going to get mad. We know a lot less about brains than certainly almost every other organ in the body. And another way to say this is that for a lot of the really important organs, we have artificial organs, artificial hearts, artificial kidneys. There's a machine that can do what the liver does, et cetera. We're nowhere close to an artificial brain. We're not even close to an artificial neuron, of which there are 100 billion in a brain. I think that's the gap that people don't realize.
What are some life lessons that have come to you through your studies in neuroscience?
So I'm not sure these are life lessons. They are life lessons. Weirdly, it's going to sound weird, but it's the truth. Learning about the brain has made me feel less guilt about my life. And guilt is in and of itself a weird emotion that I don't fully understand why we have it, how it benefits us moving forward. But now that I understand about stimulus response and I'm just a bunch of neurons, I've allowed myself to forgive some of the mistakes that I've made, because it wasn't actually like I was a person, make like a list of good things about, and then I made a decision and chose. A lot of these things only retroactively seem like decisions based on what I know about brains. And as a result, it seems like a weird thing to say.
That's interesting.
I feel less guilty about things.
Yeah.
And that's the message I give to people. Like the one thing studying brains has made me realize is that we are not in control of our actions as much as we either think or society wants us to think. So take it easy on yourself a little bit.
You had mentioned that you grew up in Goa, or spent your early childhood
in India.
there.
Yeah.
How do you think that impacted your life?
So I think what I didn't mention is that yes, I was raised by So my parents were in India, but then they moved to the U.S. when I was about 2 years old to go to school, to set up jobs, to get ready to bring me. So I wound up being raised by my grandparents for about 2 or 3 years. And as a result, I've always wanted to know how I was screwed up growing up developmentally. And maybe we were all interested in that in ways of or whatever. So,
I think so, yeah.
right. And so a lot of, when I think about why am I so obsessed with how the brain develops and et cetera, it's because these important events happen to me over development, my development. And I'm kind of curious what that means. And so I'm kind of interested in the developing brain for that reason.
This is a fascinating subject of research.
It's not well studied. Or used to be well studied. And I think it is because it's not surprising that since neuroscientists tend to be older people, we are obsessed with how much plasticity is there in the old brain? How much plasticity is there in the adult brain? Because I think for the same reason, we want to convince ourselves that this is still plastic, that this is still capable of learning. And so I really think a lot of this science, especially in neuroscience, is motivated by our own personal experiences or views of the world. I think a lot of people who are neuroscientists who study addiction, for example, because they have personal histories of one way or the other of addiction. So particularly neuroscience, I think, is this field where people say, I wonder what's wrong with me. I'm going to be a neuroscientist to find out.
Do you think intelligence could be inherited?
Do I think there's a genetic basis to intelligence? So let me start by saying I used to have a very strong opinion on this, and it was almost entirely driven by my politics. And I'll explain what that means. Broadly, in the world of nature versus nurture, I'm of the idea, and I think I still am, that it's 100% nurture.
100% nurture?
Yes.
Really?
That I used to, and I still, and I can't decide how much of that is what I know from the science, but I'll get to that in 1 second, versus how much of it is how I believe how society should be set up. Because if it's 100% nurture, then give it a good enough education system, then anybody gets to be Einstein.
Sure.
And I want that to be true. Not up here, but down here, I want that to be true. Then we got these damn results about synaptic development in mouse versus primates. And I believe that growing up a primate is a fundamentally different experience than growing up a mouse. And yet, the clocks are the same, which is really making me think there's a strong genetic program for intelligence, which is making me uncomfortable.
So then
if I'm allowed to say that.
So then you'd have to say it's less than 100% nurture, right?
It is absolutely less than 100% nurture, but I don't like saying that.
Yeah.
I don't like, despite the fact that it's our results that are making me think that, I'd still rather live in the world where it was 100% nurture. But it can't be true.
Right.
And so, sorry, that's another long answer to a short question.
No.
But yeah.
So this I read during the course of my preparations for this, but let me double check to make sure that this is correct.
Please.
It said, or I saw something that said 60% of the human brain is fat.
Huh.
Is that accurate?
Well,
is made of fat?
So I guess so. So there isn't fat cells in the brain, as far as, and we've looked at almost every part of the brain, what we call adipose tissue, like what I have in my tummy, or et cetera. Those are actually cells made of fat. But every neuron is surrounded by a membrane. And that membrane is made of something called lipids or phospholipids. So it could be that
they're calling lipids
fat. I mean, fats are lipids. And the fact that a membrane maybe accounts for 60% of a neuron, and the rest of it is the stuff inside of it is where that number comes from. But I have never seen fat cells.
Is the lipid just there for like protective reasons then?
So one, of course, it keeps things inside cells versus outside cells, but it has many other functions. It's the thing that transports receptors and channels up and down the neuron. I'm not much of a lipid biologist, but I guarantee it's critical for many different kinds of neuronal function.
How are male and female brains different?
So we have a paper that we're thinking about. No, not thinking about, that we are very close to publishing. And it's a long tangent to your answer. I'm very interested in this idea that there are sex-specific differences in brains. And just because I'm biased, I would say it's the pattern of connections in a female brain that makes it different than a male brain. But we're actually putting this potentially to the test. So in some species, there are truly profoundly sex-different behaviors. And one that we study, that many people study, is in birds. In birds, in some birds like the zebra finch, males sing. They sing these incredibly elaborate songs that's used for a courtship. In those same species, the females, they make vocalizations, but it's nowhere near as sophisticated as the male vocalizations. So we thought, okay, first we were going to look, because the brains are so big, I don't know where to look. But one place we think there's a difference is in the actual voice box. It's called the larynx in humans, but it's called the syrinx in birds. So we thought, okay, and there are connections there between the neurons in our spinal cord called motor neurons and the muscle. And it's the firing of those motor neurons that vibrates the syrinx that produces the song. So we had, and it turns out that there's this experiment you can do, which is you can give masculinizing hormones to females early on in development, and you could give feminizing hormones to males early on in development, and they wind up switching their behavior. So that females wind up singing more and males wind up singing less. So I've sort of set up the argument for you. We have this ability, and lo and behold, when we look in these muscles, we find that the kinds and types of neuronal connections are fundamentally different in the normal male versus the feminized male versus the normal female, and only in the larynx. Every other muscle is identical.
Only in the larynx.
Only in the voice box. Sorry, Paul. So that suggests that, if I could extrapolate, that many of the potential behaviors that are different across males and females is in their neuronal connections.
How has your work studying neurology impacted your humanity?
Maybe I'm repeating myself, but not only do I feel less guilt about myself, I find myself judging other humans less. Like the idea that somebody has good behavior or bad behavior, these are all just neural circuits. And a lot of those circuits were formed in that person when they were a child. And so let me try. This is maybe one of the more controversial things I tell my students. So let me try. I think it's well known that children that grow up in abusive environments, abusive families, tend to either are much more likely to become abusers themselves in their adult relationships, or be victims of abuse in their adult relationship. As a neuroscientist, here's what I believe. I believe that they are abusers of other people in adults because when they were children, they were exposed to this environment where they had no control. They didn't get to choose to be in that. They were children. And that environment caused long-lasting changes in their neuronal connections that drives their behavior as adults. So who am I to judge those people? In fact, I think the only thing I could imagine is compassion. And if that's true about these extreme behaviors, it's probably true about every jerk I've ever met in my life, including, as we were saying, Paul, sometimes the jerks who are in charge of our lives. And so if I really believe all of the things that I'm telling you, then there's very little in my mind like judgment around one politician versus one bad person versus another. I don't think of, every day I think of the world less and less as about crime and punishment or judgment, and more about healthy and unhealthy, illness and not. And I suspect that many of these a-hole traits are just inappropriate wiring diagrams, and many created during childhood. And so that's a long answer, but that's how it's impacted me.
Yeah. What is the last book that you've read, not science book, not a fiction, book of fiction? What was the last fiction book you
So I
read?
read a lot of sci-fi books, but perhaps that's not.
Well, sci-fi, that would work, yeah.
Yeah, I mean,
It's-
and I think now it's a TV show on Netflix, but the Three Body Problem, which is a sci-fi book. Have you?
no, I'm not familiar with it.
Amazing. So, just if you have 2 minutes,
Yeah.
I always recommend this book. First, it's in China. It's a Chinese sci-fi author, and in China, it's considered almost beyond sci-fi. It's considered literature, the equivalent of maybe what we would say Shakespeare or et cetera. And it is an entirely different view of sci-fi than I've ever met. I grew up on Western Isaac Asimov, et cetera. And in Chinese sci-fi, at least in this book, there are very few cowboy heroes. In all of the literature, sci-fi literature, there's always one, usually guy, who saves the day. Chinese sci-fi it's much more realistic than that, and much more epic in scope. And that's one of the things I found fascinating. And it never occurred to me, of course it's true, that the literature of a particular country is a reflection of the country's worldviews, obviously. Yeah. But that is true about sci-fi as it is about every other romance or mysteries or whatever. Yeah. And I was like, holy crap, you could back out what a society thinks by reading enough of its literature. So that's the last one. And also, and I don't want to do a spoiler alert, but man, it is a book about first contact, and I've never. You should read it. I don't want to tell you, but it explains this thing that I've been struggling with for a long time, which is called Fermi's paradox, which is if there are so many planets and stars out in the universe, and the universe has been around for so long, where are all the goddamn aliens? And this is the best explanation of the Fermi paradox I've ever heard. And it was one of those moments where it just blew my mind. Should I tell you, or?
Sure, yeah.
And it helped me think about the world as well. And I'm sorry to the viewers who haven't read it, so this is the spoiler alert moment. The idea of the book is that all civilizations on Earth, but also in space, aliens, either grow or die. So maybe that's a reasonable assumption. And the second assumption is the amount of matter and resources in the universe is finite. And so it means that the only successful civilizations out there, it's called the dark forest analogy, which is that the universe is filled with civilizations, but none of them want to reveal themselves, because as soon as they reveal themselves, every other civilization attacks them and takes their resources. And I was like, holy cow, that's probably the answer. And then every now and then I think, I hear the government is like, we're going to send out another SETI, and I'm like, no, 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 no. Let's not tell anybody we're here. Let's keep it as quiet as possible until we're advanced enough to do. Anyway, I mean, and that's the most recent non sci-fi book I tried last year is that I tried to reread Moby Dick.
I just reread it a couple of years ago.
And and I realized that my English teacher did me a favor because we only read the non-shipping chapters. Like there's an abridged Moby Dick apparently.
That would take out like 3/4 of the book, wouldn't it?
And so somebody told me that, and I was like, oh, I got gypped, you know, like, and then I was like, so I'm going to reread the damn. Holy cow! That is not easy. The amount of detail about whaling and shipping is profound. And so I couldn't make it through. I got lost in the whaling. But now that I heard you did it, I'm going to try it again.
You should.
Is it worth it?
I think so.
Okay.
Yeah, you just have to look up half those things.
Right. Exactly.
That's what I did.
Okay. And I want to reread Heart of Darkness, this Conrad book. I remember reading it when I was young, in high school, and I remember it having a profound impact on me. And then only decades later did I see the movie,
Apocalypse Now.
Apocalypse Now. Yeah. And man, that movie is one of those moments where I just blew my mind. And it was only like maybe 10 years ago I saw the movie, finally. And I was like,
you should see the documentary about the making of it, too.
No kidding.
Yeah.
No kidding.
Yeah.
Because it was crazy. What's his name had a heart attack. The other guy was on drugs the entire time. Right. Dennis Hopper, maybe.
All kinds of stuff. Yeah.
Right? And so, so it really made me want to then reread the book. And also because I've been thinking, and I read an essay later on by a very famous Nigerian author who I love, named Chinua Achebe. He wrote a book called Things Fall Apart, several Man of the People, and I remember reading those books and just, again, a cultural, the idea that this is the kind of literature of that region of the world, and you could learn so much about it. But then Achebe wrote a book, an essay, much later on, about was Conrad racist? Is Heart of Darkness racist? Meaning the depiction of Africa, the depiction of the savages, the depiction of, is it Kurtz or whatever, the main actor who gets, you know, taken in by this culture. And, you know, of course, his last words are, the horror, the horror. And I guess there's been English PhD after English PhD about who is he talking about? Is he talking about the Africans? Is he talking about himself? Is he talking? And so I don't know why, I really wanted to reread Conrad again to see if I got suckered. Like, is this book that was so fundamentally important to me? And I don't even know, it just seems important to me. Was it racist in ways that I didn't understand? So I have a list of future books, I guess, is a long answer to your short question.
What's one piece of advice you'd like to share with the listeners, viewers?
Geez, I don't know why anybody would take advice from me on anything, but I don't know.
Well, how about advice for someone that says, hey, I want to be a neuroscientist?
Yeah, okay, great. I would say that this is not something I realized when I said I wanted to be a neuroscientist. That is that, and again, Paul, just because I always have stories and questions for students, sorry, not kids, students, sorry. And so it's the only way I can think about these things. So I'll give you the advice I give them. So I say to them, and if you don't mind, I'll ask you, I say, what is the purpose of science? And then I tell them, like I'm going to tell you, don't worry, I've never heard the right answer to this question, and so almost any answer you're going to give, I'm going to say is wrong. And this is a lesson that I would tell every young scientist. The purpose of science is to make more scientists. And in fact, I would argue the purpose of anything, music, the law, medicine, is to make more musicians, artists, lawyers, doctors. Because if something is truly worth doing, then it's worth doing out into the future, right? So that's the first thing I say. And then the second thing I say is, I don't know where we got this idea. It was taught to me, or in a not obvious, like in the background, it was taught to me that there are biological questions that get answered in a single generation. And I have never encountered one as I got older and older and older. Nothing ever gets solved in a single generation. Yeah. So, but yet we live these lives and where we imagine that I'm going to become a scientist, I'm going to answer a whole bunch of profound, important questions, and we're going to understand the subject much better in a single lifetime, which is nuts. And in fact, what I tell students is that science is much more like how they used to build cathedrals. This is another book I read. I read this book, Giants, Pillars of the Earth or something, which is a fiction book about cathedral building in Europe in the 12th whatever century. And what again blew my mind is, of course, every cathedral built takes multiple generations to build. So how is Christopher Wren, let's say, some famous architect of cathedrals, how does he know? How does he ensure that that cathedral, which is going to be, not even get started till after he's dead, is going to be built the right way? The way he does it is the way we do it. We train a bunch of apprentices, and we teach them we try to convince them that our view of the world, our view of science, is the right way to do it. Flying buttresses, please, not whatever, 4 years or whatever. And if we're successful, they adopt our worldview and they continue to teach. And then that's actually how you get your science done. And I would say the window that I learned over which you actually contribute to science, as opposed to you contributing to making more scientists, that first window is extremely small. And you have to be extremely lucky, I think, to nail it in that window. So for most people, not him, most of science is about training the next generation of scientists. And I feel like that's the life lesson it really has taken, beat into me. Because I had imagined I was so smart and so hardworking and so dedicated that it would be the former. And I realized that the real payoff is the latter. And it's the long range payoff. And if you come in knowing that that's the real payoff, then science is like no other field I've ever been in. Because the ability to nurture or to work with the next generation is amazing. The ability to actually discover things about stuff is incredibly frustrating.
It's great advice. Yeah, I like that.
And so that's it. And I think that's true about any field. Whether you're a lawyer, like the actual moments of cases that are gonna profoundly, Supreme Court, small. But the idea of training the next generation of lawyers in your view of what you think makes a great lawyer is a real and achievable impact. And I think that's my advice.
Well, thank you, Bobby. It's been a pleasure.
Wow, it was really amazing, Paul.
We'll have to do it again.
Anytime. Anytime.